Thank you. Today I'll be speaking about the critical role of high-quality human biological specimens in advancing research and personalized medicine. Biomarkers are the backbone of personalized medicine, and BioIBT's mission is to enable smarter science and accelerate medical breakthroughs that enhance and extend lives by delivering personalized biospecimen solutions to life science and diagnostic industries. Last year, I sat down with our team and went through the estimated cost of using poor quality biospecimens in one's research. As can be seen here, it can easily add up to over $30,000 in hidden costs due to biospecimen failure. I will go through this in this talk, some of the key steps that you should take, and key questions you should ask of your biospecimen providers. This talk has been divided into 4 sections: true cost, key considerations, preserving sample integrity, and ensuring reproducibility and innovations in the future of personalized medicine. First, let's dive into the true cost of poor quality specimens and why quality matters. A survey of drug discovery researchers found that over 60% of the researchers questioned their findings due to lack of quality biospecimens. In addition, this led to an 81% limiting the scope of their work. Factors affecting biomarker translation include pre-analytical variability from poor sample handling, inconsistent SOPs, non-standardized tube types, and more, missing clinical metadata including age, race, gender, as well as clinical treatment, comorbidities, family history, and more, and assay reproducibility gaps. The hidden costs come from different factors, and these include re-embedding, which can be 25 to 100 a block, failed assays at $150 to $500 plus reagent cost, diagnosis confirmation, which can cost $150 an hour for pathology to reassess each block, replacement specimens from 300 to 2,000, depending on the rarity of the specimens. And then there is the cost of the days of delay. As preclinical R&D costs are estimated at $3,000 per day, these can add up. As you can see, histology processing can take 2 days or $6,000. Assay time, including troubleshooting, can take 7 days or $14,000. Ordering specimens can take 7 days or $14,000. And as you can see, the costs add up quite quickly. What are the key things that we need to look at when it comes to high quality? There are things such as the histology quality control, which ensures appropriate size criteria, specimen integrity, is it free from bubbles, discoloration, cracking, contamination, and the use of a quality scoring system in the quality of the FFPE blocks. Pathology and data into review, board-certified pathologists confirm diagnosis, tumor grade, and stage with discrepancies leading to quarantine. Ethical oversight includes documented patient consent, IRB approvals for broad research use, external quality assurance, and annual auditing of clinical sites. At BioIBT, we have a 30-year history of following all of these procedures. Now I will go through the key considerations for selecting an HBS provider and questions that you should ask your providers. First, we have regulatory and quality requirements. Does the provider have the expertise and resources to meet all necessary regulatory requirements? Next, we have a strategic partner network, or SPN. Does the provider have a proven SPN capable of servicing your specific human biological specimen needs? SPN evaluation. How is the human biological provider's strategic partner network evaluated, contracted, and audited for compliance? Donor recruitment. Does the provider have the ability to create a dedicated donor recruitment process for your HBS needs? Continuing, there is supporting regulatory documents. Can the provider provide supporting regulatory documents such as blinded consents and IRB approvals for your regulatory submissions? Consistent quality products. Does the provider have procedures in place to deliver consistent quality products? A quality management program. Does the provider have a QM program in place? A chain of custody policy. Does the provider have a chain of custody policy in place? Pathologist certification. Does the provider certify specimen type and diagnosis with a board-certified pathologist to verify diagnosis? Independent clinical data review. Does the provider have an independent individual review of the clinical data which is then corresponded with the pathology certification of that diagnosis. Customized collection. Does the provider have the ability to customize collections while maintaining regulatory and quality standards? And finally, but certainly not least in my eyes, is overcoming challenges. What challenges has the provider overcome for clients in the past? To me, this is one of the most critical questions that you should ask every human biological specimen provider you plan on working with. Every provider has a time where there was a challenge to them. And talking about how they overcame those challenges with logistics, with identifying a donor cohort, et cetera, is critical to understanding how they can work with you to help you be successful. So next we'll talk about preserving sample integrity and ensuring reproducibility. All of the previous questions go to support this in support of your research. Again, essential protocols need to be in place for sample integrity for biomarker discovery. These include histology quality control to ensure appropriate size criteria, specimen integrity, and using a quality scoring system, ensuring that the procedures are regularly reviewed and updated to meet industry standards. Pathology and data integrity review. Board certified pathologists confirm diagnosis, tumor grade, and stage of every block, with discrepancies leading to quarantine of those blocks. For cancer-related human biological specimens, they should determine the parameters such as percent tumor, necrosis, inflammation, and normal regions within each tissue block. Ethical oversight includes documented patient consent, IRB approvals for broad research use, external quality assurance, and annual auditing of clinical sites. Standardized collection procedures, rigorous, consistent protocols for collection methods, container types, processing times across all sites. Rapid processing and storage, samples processed within 4 hours with biofluids centrifuged, aliquoted into smaller volumes, and stored at -80 degrees to prevent degradation and minimize freeze-thaw cycles. Detailed sample annotation, comprehensive documentation including collection date, time, processing steps, storage conditions, and any deviations. Quality control measures, regular QC checks such as protein concentrations or RIN analysis, standardized reagents, internal QC samples, and external quality assessment programs. Minimize pre-analytical variations with control factors like fasting status and time of day for sample collection should be recorded. For the researcher, ensuring uniformity and reproducibility across collections includes batch effect mitigation, randomization of samples across batches, inclusions of QC samples and statistical correction for batch effects, training and certification, comprehensive training for all personnel involved in sample handling and analysis, and use of high quality repositories, utilizing well-characterized samples from reputable biospecimen repositories. And when all these standard collection protocols, processes, auditing, reviewing, this leads to adopting the industry's best practices with organizations like the Human Tissue Authority in the UK and the College of American Pathologists in the US, establishing standards. BioIVT is committed to adhering to these new standards as they are established. If we look at emerging innovations in biospecimen and biomarker validation, we have the integration of AI and machine learning, which revolutionizes biomarker analysis by processing large data sets, identifying novel biomarkers, and predicting treatment responses. Advanced analytical techniques, such as mass spec, next-gen sequencing, high-resolution microscopy, all providing deeper insights. Techniques like LC-MS/MS and mesoscale discovery, or MSD, offer superior sensitivity and multiplexing capabilities. Liquid biopsy and non-invasive testing, such as the shift towards analyzing circulating tumor DNA or ctDNA in blood for earlier detection and monitoring. Digital biomarkers emerging as new frontiers, such as AI-driven platforms for neurological disease and diagnosis through eye movements. Blockchain technology, revolutionizing traceability, security, and authenticity in real-time specimen tracking. And robotics and Internet of Things, streamlining sample collection, storage, and real-time monitoring of conditions and locations. BioIVT supports all new innovations and has implemented the following future-forward strategies. expanding service offerings such as our recently introduced Ki-67 tissue staining service for antibody drug conjugate and radioligand development. Leveraging advanced technologies, we utilize Aperio full slide imaging and next-gen sequencing in-house to support enhanced analytical capabilities. Focused on high demand research areas, we launched the Center for Neuroexcellence for neurodegenerative disease studies such as Alzheimer's, Parkinson's, and ALS. Extensive repository we manage the largest private global repository of longitudinal collected human cerebral spinal fluid, CSF, samples, as well as large tissue and biofluid repositories to support cancer, autoimmune, and other research areas. Strategic acquisitions with acquisitions of companies like Precision Med, SynBio, and Xenotech to provide expanded solutions in developing areas such as exosomes and ADME-Tox consulting, and a collaboration and customer-centric policy. We established a scientific advisory board of industry experts, and we regularly gather customer feedback to align quality controls with needs. And this brings me to the end of our talk with one final statement. High quality human biological specimens are not a luxury, but a necessity for accurate, reproducible, and impactful research. Choosing an HBS provider with rigorous quality control, ethical practices, and comprehensive infrastructure is paramount. The field is rapidly advancing with exciting innovations that promise to support the revolutionizing of personalized medicine, and BioIVT continuously invest in innovation, quality, and ethical practices to support the advancement of science and accelerate medical breakthroughs.